Welcome to today's lecture on the intricate and fascinating topic of carbohydrate metabolism. Carbohydrates serve as a major energy source in our body, playing crucial roles in various physiological processes. In this lecture, we will delve into the detailed regulation of carbohydrate metabolism, exploring the enzymes and hormonal factors that influence this essential biological pathway. We will also examine the dysregulation of carbohydrate metabolism in diseases like diabetes mellitus, as well as the impact of different medications on its regulation. So, let's begin our journey into the world of carbohydrate metabolism. Carbohydrate metabolism is regulated by an intricate network of enzymes and hormones. One of the key enzymes involved is hexokinase, which converts glucose into glucose 6-phosphate in the first step of glycolysis. This enzyme is allosterically inhibited by its product, glucose 6-phosphate, providing a negative feedback mechanism to regulate glucose utilization. Additionally, glucokinase, a liver-specific isoform of hexokinase, plays a crucial role in maintaining blood glucose levels. Hormones such as insulin and glucagon exert profound effects on carbohydrate metabolism. Insulin, released by the pancreas in response to elevated blood glucose levels, stimulates glucose uptake by tissues and promotes the storage of glucose as glycogen. It also suppresses gluconeogenesis, a process in which glucose is synthesized from non-carbohydrate precursors. On the other hand, glucagon, released during periods of low blood glucose, stimulates gluconeogenesis and glycogenolysis, leading to an increase in blood glucose levels. Glycolysis, the multi-step process by which glucose is converted into pyruvate, is tightly regulated. One critical regulatory step is catalyzed by phosphofructokinase 1, PFK1. PFK1 is allosterically activated by fructose 2,6-bisphosphate, an intermediate formed in response to high insulin levels, while it is inhibited by ATP and citrate. This allosteric regulation allows for the fine-tuning of glycolytic flux according to cellular energy demands. Furthermore, the enzyme pyruvate kinase, which catalyzes the conversion of phosphoenol pyruvate to pyruvate, is regulated by hormonal factors such as insulin and glucagon. Insulin activates pyruvate kinase, favoring glycolysis and glucose utilization, while glucagon inhibits this enzyme, promoting gluconeogenesis. Gluconeogenesis, the process by which glucose is synthesized from non-carbohydrate precursors, occurs predominantly in the liver and plays a crucial role in maintaining adequate blood glucose levels during fasting or prolonged exercise. The key regulatory step in gluconeogenesis is the conversion of oxaloacetate to phosphoenol pyruvate, catalyzed by the enzyme phosphoenol pyruvate carboxykinase, PEPK. Hormonal control also governs gluconeogenesis. Glucagon stimulates the expression of gluconeogenic enzymes, promoting glucose synthesis, while insulin inhibits gluconeogenesis. Moreover, cortisol and certain catecholamines can enhance gluconeogenesis by increasing the availability of gluconeogenic precursors, such as amino acids. Glycogen, a branched polymer of glucose, serves as a rapidly mobilizable storage form of glucose. Its synthesis and breakdown are tightly regulated to maintain glucose homeostasis. Glycogen synthesis occurs via the enzyme glycogen synthase, which adds glucose units to the growing glycogen chain. The activation of glycogen synthase is regulated by phosphorylation. Protein kinase A, pKa, activated by glucagon signaling, phosphorylates and inactivates glycogen synthase, preventing glycogen synthesis. In contrast, insulin stimulates glycogen synthesis by activating protein phosphatase 1, PP1, which dephosphorylates and activates glycogen synthase. AMP-activated protein kinase, AMPK, acts as a master regulator of cellular energy status. It is activated under conditions of low energy, high AMP/ADP to ATP ratio, and acts to increase energy production while inhibiting energy-consuming processes. AMPK inhibits anabolic pathways, such as protein synthesis via its effects on the mammalian target of rapamycin, mTOR, signaling pathway. The mTOR pathway integrates nutrient and energy signals to regulate cellular processes such as protein synthesis, cell growth, and proliferation. mTOR activation promotes glycolysis and inhibits autophagy, contributing to cell growth and survival. Dysregulation of AMPK and mTOR signaling has been implicated in various metabolic disorders, including obesity, type 2 diabetes, and cancer. In response to changes in nutrient availability and energy demands, our cells can dynamically switch between different fuel sources. During fasting or prolonged exercise, the body adapts to utilize alternative energy substrates, such as fatty acids and ketone bodies, to meet its energy needs. This metabolic flexibility allows for the conservation of glucose for the brain and red blood cells. The shift from glucose to fatty acid utilization is coordinated by hormonal regulation. Hormones like glucagon and catecholamine stimulate lipolysis, releasing fatty acids from adipose tissue, while insulin inhibits lipolysis, favoring glucose utilization. The complex interplay between these hormonal signals and enzymatic regulation ensures a tight balance between glucose and fatty acid metabolism. The maintenance of blood glucose levels within a narrow range is vital for normal physiological function. The liver plays a central role in this regulation, constantly adjusting glucose production and uptake to match the body's requirements. During fasting, glycogenolysis and gluconeogenesis are upregulated to produce glucose, while in the postprandial state, insulin suppresses hepatic glucose production and facilitates glucose uptake by tissues. The coordinated action of hormones, such as insulin, glucagon, cortisol, and growth hormone, ensures the precise regulation of hepatic glucose metabolism. Dysregulation of these hormonal mechanisms can lead to clinical conditions like hyperglycemia and hypoglycemia. Exercise poses a substantial metabolic challenge to our body, demanding an increased supply of energy substrates. During short bursts of intense activity, glucose serves as the primary fuel source, derived from both glycogen breakdown and circulating blood glucose. As exercise duration extends, muscle glycogen stores become increasingly important. Regular exercise training induces adaptations in carbohydrate metabolism, including increased muscle glycogen storage capacity, enhanced glucose uptake, and improved insulin sensitivity. Understanding the intricate regulation of carbohydrate metabolism during exercise can aid in optimizing athletic performance and managing metabolic disorders. Diabetes mellitus is a complex metabolic disorder characterized by impaired glucose homeostasis. In type 1 diabetes, the body fails to produce insulin, leading to a disruption in carbohydrate metabolism. In type 2 diabetes, insulin resistance develops, impairing the ability of tissues to respond to insulin. These abnormalities in insulin signaling and glucose metabolism have far-reaching consequences. Chronic hyperglycemia can result in the development of various complications, including cardiovascular disease, neuropathy, and nephropathy. Understanding the underlying molecular mechanisms of carbohydrate metabolism dysregulation in diabetes is crucial for developing effective therapies. Glycogen storage diseases, GSDs, are rare inherited disorders characterized by defects in enzymes involved in glycogen synthesis or breakdown. These enzymatic defects disrupt glycogen metabolism, leading to abnormal glycogen accumulation or depletion. Different GSDs manifest with varying clinical presentations and severity. For example, in GSD type I, also known as von Gerke's disease, a deficiency in glucose 6-phosphatase impairs glucose release from glycogen in the liver and kidney, resulting in severe hypoglycemia. Understanding the underlying enzymatic defects in GSDs aids in diagnosing and managing these challenging disorders. Carbohydrate metabolism is regulated differently in various tissues to meet their specific energy requirements. For instance, the brain relies heavily on glucose as its primary energy source and utilizes ketone bodies during starvation. In contrast, skeletal muscle can switch between carbohydrates and fatty acids based on energy demands. Tissue-specific regulation involves the orchestration of unique enzymatic activities, hormonal signaling, and substrate availability. Understanding these tissue-specific adaptations provides insights into the physiological and pathological processes related to energy metabolism. Numerous medications can influence carbohydrate metabolism, either directly or indirectly. For example, certain antidiabetic medications promote insulin release, enhance insulin sensitivity, or inhibit gluconeogenesis, thereby aiding in the management of diabetes. Other medications, such as glucocorticoids, can induce insulin resistance and disrupt glucose homeostasis. Understanding the impact of medications on carbohydrate metabolism helps clinicians make informed decisions when prescribing treatments and manage medication-induced metabolic disturbances. In conclusion, carbohydrate metabolism is an intricately regulated process that ensures a constant supply of energy for cellular functions. The interplay between enzymes, hormones, and signaling pathways governs glucose utilization, storage, and production, maintaining optimal blood glucose levels. Dysregulation of carbohydrate metabolism can lead to metabolic disorders like diabetes mellitus, glycogen storage diseases, and disruption of cellular energy balance. By gaining a comprehensive understanding of the regulation and dysregulation of carbohydrate metabolism, we can develop strategies for managing metabolic disorders and promoting overall health and well-being. Thank you for joining this comprehensive lecture on carbohydrate metabolism.